So who can remind us what are the 3 ways to phosphorylate to make ATP? What are the 3 types of phosphorylation?
Substrate level phosphorylation.
Yep.
Oxidative phosphorylation and
Yep.
photosynthesis.
The last one?
Photosynthesis?
Well, photosynthesis, using the process of photosynthesis. So the idea is to phosphorylate ADP to make ATP. And so there are 3 phases or stages of carbohydrate catabolism. And what's the primary carbohydrate we're talking about here? What's the number 1 carbohydrate that's used for energy?
Glucose?
Glucose, right? Remember glucose? Well, there are 3 steps or stages or phases of glucose metabolism: glycolysis, the Krebs cycle, and electron transport chain. So a glycolysis or glycolysis is the splitting of the sugar glucose. The Krebs cycle, also known as the citric acid cycle, we'll review, and then the electron transport chain and chemiosmosis. So you should recall the equation for aerobic cell respiration. So what's the equation for aerobic cell respiration? What's the formula for glucose? That's a formula you should all have memorized. Glucose. Roxy, do you remember? Christina? Okay. C6H12O6, right? And then what do you need to break down that glucose or to oxidize it or to burn it? What do you need? Oxygen, right? How many? 6. And then that yields what?
Carbon dioxide.
Carbon dioxide, how many?
6.
6. Plus what?
Plus 6 H2Os.
Yep, 6 waters. Plus what?
And then ATP.
Is that all? What other form of energy? No, not NADH. A form of energy. One that's very important, especially in the wintertime.
Heat.
Heat. So C6H12O6 is oxidized using oxygen to yield CO2, water, and 2 forms of energy, ATP and heat. Which form of energy is greater, ATP or heat? I got 2 ATPs. It's actually heat. More heat is generated from the original chemical potential energy in the bonds of glucose. only about 40% is stored as ATP. So we're going to talk about the oxidation of food molecules like glucose to liberate the electrons for an electron transport chain. Because as we'll see, it's all about the electrons. And the overwhelming majority of ATP will be generated by oxidative phosphorylation. Now, we're not going to go into all the same detail that we did in Biology 150, but the main idea is to focus on what gets made, how much of it gets made, and where it gets made. Because we are going to compare and contrast eukaryotic cells with prokaryotic cells. But we start off with glucose, and glycolysis is a universal pathway. So basically all the forms of life on the earth can do this. And by the end of glycolysis, that 6-carbon glucose is going to be split into 2 3-carbon pyruvic acids. In the process, some ATP will get made as well as some NADH. Now, what can you tell me about money? If you want to make money, what do you need? A bank. Not necessarily. Banks take your money. So what do you need? A job. But how does the old expression go? If you want to make money, what? Well, you need money, right? So you need to invest money to make money. Same idea with ATP. In order to make more ATP, you have to invest some ATP to start with. And so that's why the cell must have some ATP in order to make more. If the cell has no ATP left, it's going to die. So 2 ATPs are used to jumpstart the process. So they're used to initiate the process of glycolysis. Like I said, we're not going to worry about all these intermediates here. You need 2 ATPs to start the process. By the time you're finished, technically, at the end of glycolysis, 4 ATPs have been made, but since the cell had to burn 2 ATPs to start, we say the net production is just 2 molecules of ATP. So we talk about the production of ATP during glycolysis to be 2 molecules of ATP. The cell also makes 2 molecules of NADH. Who can tell me, what are the NADHs carrying?
Electrons.
Electrons, right? So we've split our 6-carbon glucose. We now have 2 3-carbon pyruvic acids, also known as pyruvate. So we broke one of those covalent bonds, and we captured those electrons in the NADH. So glycolysis occurs where in all cells?
Is it outside of the mitochondria?
Right? So,
Oh.
but no, I mean, you're right, but what would you call that?
In the cytoplasm?
In the cytoplasm, so all cells, right? Prokaryotes, because they don't have mitochondria. So glycolysis occurs in the cytoplasm. Now, before the Krebs cycle can begin, there's a problem with the pyruvic acids. They cannot directly enter into the Krebs cycle. They must be converted. They must be decarboxylated first into acetyl coenzyme A, or acetyl-CoA for short. Here we see another carbon comes off. So another covalent bond has been broken. We release that carbon and capture the electrons in the NADH. So where are the NADHs going to carry those electrons? The NADH is carrying electrons to where? They got to go somewhere. So where do they go? So the electrons are carried to the electron transport chain for the purpose of making what? ATP, right? ATP. Now, how many molecules of NADH are produced in this intermediate step? I know it sounds like a simple question, but how many NADHs are produced here at this step?
Is it 2 because there's 2 pyruvic acid molecules?
That is correct, right? So there are 2 pyruvic acids, so each one will go through and be decarboxylated to produce acetyl-CoA. So 2 NADHs are produced, as well as 2 CO2s get released. So now we're ready for the Krebs cycle. So if we're in a eukaryotic cell, where are we now? In a eukaryotic cell?
So what we just talked about wasn't the Krebs cycle, or it was?
Not yet, that was an intermediate step. Right, the pyruvic acids must be converted into acetyl coenzyme A. Now the acetyl-CoAs can enter into the Krebs cycle. But where are we now in a eukaryotic cell? Inside the, specifically the mitochondrial matrix, but we're inside the mitochondrion, right? Where are we in a prokaryotic cell? Where are we in a prokaryote? Do prokaryotes have mitochondria? We're in the cytoplasm, that's right, Karine. So for prokaryotes, we're still in the cytoplasm. Eukaryotes, we're in the mitochondrion. So our acetyl-CoA is going to go through all these different intermediates, which fortunately for you, we don't have to know. But in this process, all the remaining carbons are going to be ripped off and released as carbon dioxide. So the original glucose, every carbon one by one gets ripped off and released as CO2. When the covalent bonds are broken, the electrons are going to be captured, in particular by NADH. So how many NADHs are produced here in Krebs? Six, four, two. Well, we have 1, 2, 3, and this happens twice, right? So the correct answer is 6. How many ATPs are made here?
It's 2 total.
Right, so only 2 are made in Krebs, right? Because remember, this happens twice, so just 2. So how many ATPs has the cell made since the beginning? And we are talking about the net production, but how many ATPs since the beginning?
Is it just 1? 2? Sorry.
How many ATPs since the very beginning? How many in glycolysis? The net production from glycolysis, how many? Well, Gareen, is that the previous question or the new question?
I meant because isn't there 2 in glycolysis and then 2 in Krebs cycles? So
Yep, correct. So a total of 4, right? So Look at all the effort the cell has gone through, all those steps in glycolysis, all these steps in Krebs, all for 4 lousy stinking molecules of ATP? That's a lot of work for just 4 molecules of ATP, right? That's not very good. But what has the cell made a lot of? NADH. How many NADHs since the beginning? N, right? We made 2 in glycolysis, 2 in the intermediate step, and 6 during Krebs. That's 10 molecules of NADH. And what are they carrying? Electrons to where? The electron transport chain to make what? ATP, right? So it's all about the electrons. Now, in Krebs, something else also gets made. We have FADH2. How many molecules of FADH2? 2. So that's a total of 12 electron carriers. So that's a lot, right? So the cell, it ripped apart those that glucose molecule carbon by carbon, a lot of biochemical reactions, only 4 molecules of ATP, but 12 molecules of NADH and FADH2. So 12 electron carriers. And all those electrons are going to be taken the electron transport chain to make ATP. So the cell has stored a lot of potential energy in those electrons, in those electron carriers. So that's the whole purpose of glycolysis and Krebs, is to rip apart the glucose, capture the electrons, and then use the electrons to make ATP. Any questions?
So FADH is also an electron carrier.
Yep.
Does it have, I mean, I'm assuming it has different properties, but it mainly just does the same thing as NADH.
Yeah, pretty much. I mean, it has other functions inside the cell, but here we're just concerned about its function as an electron carrier.
Okay, got it.
So the electron transport chain, a series of carrier molecules that are oxidized and reduced as the electrons are passed down the chain. So when the electrons are donated to the first carrier complex, it gets reduced, and then the electrons are passed on to the next complex, and the next complex gets reduced, and the previous complex is now oxidized, and so on and so forth. So oxidation, reduction, oxidation, reduction. Each time the electrons are passed from complex to complex, their energy is lowered, it's reduced, and those complexes also transport hydrogens across the membrane. And we'll call that process the chemiosmosis. So here we see an electron transport chain. The NH is donating to the first complex in the chain. That causes the complex to get excited and transport a hydrogen across the membrane. As the complexes are transported, or as the electrons are transported from complex to complex, it causes more hydrogen to get pumped across the membrane, and they accumulate a high concentration. What do you know about molecules in a high concentration? Where do they want to go?
An area with lower concentration.
To an area with lower concentration, which is back on the other side of the membrane. But the hydrogens cannot simply diffuse through the membrane, so how do they get back to the other side? right? The hydrogens are in a high concentration on one side of the membrane, and they want to get back to the other side where the concentration is low, but they cannot simply diffuse through the membrane. So how do they get back through?
An ion pump?
Not a pump, right? Pumps typically require energy, but just in general, how do molecules get to a membrane if they can't diffuse?
Oh, like a channel, through a channel?
Right, a channel through a transport protein, right? And it can be passive transport, which it is in this case. So that transport protein is no ordinary transport protein. It's actually an enzyme, ATP synthase. So the hydrogens flow back through the ATP synthase, following their concentration gradient from high to low, and the physical movement of the hydrogens is a form of energy that ATP synthase uses to generate ATP. Because at this point, ATP synthase cannot use ATP to make more ATP, right? Because making ATP requires energy, and that source of energy cannot be ATP because there would be no net gain, right? So there needed to be an alternate form of energy for the cells to use. That alternate form of energy is a hydrogen ion gradient, also known as by a fancy name, the proton motive force. So the proton motive force, because hydrogens are basically just a proton, right? Just one proton circled by one electron. So it's just a proton. So a concentration gradient is formed. And those hydrogens flow back through ATP synthase from their concentration being high into an area that's low, and they go through the enzyme ATP synthase. And so that physical movement of hydrogens is an alternate form of energy that ATP synthase uses to make ATP. So that's pretty amazing, right? You should all say, yes, that's amazing.
It's amazing. So just by flowing through back into the, I guess, inside to the space,
Yep,
it makes energy just by going
yep,
inside.
yep. So it's kind of through a dam and rotating turbines. It's that same idea. The physical movement of the hydrogens is an alternate form of energy that allows ATP synthase to make ATP. Even more amazing, in order to pump the hydrogens, remember that when cells create a concentration gradient, so when they pump substances against their gradient to concentrate them, that requires energy. And so again, here the cell cannot use ATP to pump hydrogens, otherwise there'd be no net gain of ATP. So the alternate form of energy used to pump hydrogens are the electrons. And the energy associated with the electrons as they pass down the chain, energy gets released. And that energy is used to pump the hydrogens, which then flow through ATP synthase. So we actually have two forms of energy here, electrons and a concentration gradient. And that's how cells make ATP. And it's the same basic idea for all cells. It works the same kind of way in photosynthesis. A hydrogen ion gradient is created, and those hydrogens flow back through ATP synthase. And that allows cells to make ATP. So it all is pretty amazing when you think about how something like this could possibly have evolved. But there needed to be alternate forms of energy to make ATP. So the electrons and the hydrogen ion gradient are used to make ATP. So it is pretty impressive when you think about it. So in this image we're looking at, what type of cell are we looking at here? A eukaryote or prokaryote?
Eukaryote.
a eukaryote, right? Because we see intermitochondrial membrane. And so the question, why are we looking at a eukaryotic electron transport chain? And this is a course on microbiology and bacteria. Why are we not looking at a bacterial electron transport chain? And the reason why is because all these fine details, especially the cytochromes, are different from species to species or genus to genus in prokaryotes. So there's no one model that perfectly replicates all the prokaryotic electron transport chains. So we use the eukaryotic version as the standard because it's the same for all the eukaryotes. And the overall principle is exactly the same. It's just that for prokaryotes, some of these fine details change. The exact same idea. Now, you do recall in eukaryotes, this is all happening inside the mitochondrion, and the inner mitochondrial membrane is very highly folded. Why is it highly folded? What's the benefit of that? More surface area, because you have all these transport chains packed in the membrane. So the more membrane you can pack into that mitochondrion, the more energy that can be produced in a very small volume. So now this process is called aerobic cell respiration. Why is it aerobic?
It uses oxygen.
Yes, it uses oxygen. So what's the purpose of the oxygen? So what's the function of oxygen in aerobic cell respiration? Nobody knows what the purpose of oxygen is? I know you all learned it. Well, how does it allow the process to happen, Christina? I mean, it does, but how?
Does it need the oxygen to be able to break the CO2s into take it?
No, not really, but you see it in this diagram here. What's the function of the oxygen?
Oh, to make the water?
Well, that's not the function to make water, that's what happens, but the actual function is to pick up those electrons at the end of the transport chain. When the electrons reach the end, they have to go somewhere, right? They can't just float around through inner space. And so the electrons combine with the oxygen to make water. So water is a metabolic byproduct of cell respiration. It's not enough to live on, but it's a metabolic byproduct. But oxygen has to be there to pick up those electrons. That keeps the electrons flowing, which keeps the hydrogens pumping. If there's no oxygen, then all the complexes get full of electrons and everything stops. No more hydrogens get pumped. No more ATP gets made here. If you're a single cell microorganism, that's okay. You can live off of glycolysis alone, but humans and other complex multicellular animals cannot. We need to make a lot of ATP. So when you stop breathing, you have 4 to 5 minutes before your brain starts to die. So your brain dies first. And that's because despite what you see with some people, our brains use a tremendous amount of ATP. In fact, our brains consume the majority of the glucose that we eat every single day. So brains require a tremendous amount of ATP, and you need oxygen for that. So if you stop breathing or you're deprived of oxygen, you got 4, to 5 minutes usually before brain damage starts to occur. So that's why you need to breathe. You need the oxygen to be the final electron acceptor in the electron transport chain, so your brain can make lots of ATP. Oh, one more question. So we know that hydrogens are pumped in the space in between the inner and outer membranes of the mitochondria. Where do the hydrogens get in a prokaryotic cell? Right? Because they don't have mitochondria, so all their transport chains are in the plasma membrane, the cell membrane. So then are the hydrogens pumped to the outside of the cell, or are they pumped to the inside of the cell? I have Christina saying they're pumped to the inside. Anybody agree or disagree? Agree, Roxy agrees. So think of H+. What does it mean if you have a high concentration of H+? Do you remember a concentration of H+? Right, the hydrogen ions. If you got a lot of them, it's more acidic, right? So do you think it would be good for the bacteria to pump hydrogens to the inside of the cell? Yeah, not really. So in fact, they actually pump the hydrogens to the outside of the cell. In gram-positives, there's a little space between the membrane and the thick peptidoglycan layer. And in gram-negatives, you have the periplasmic space in between the two membranes. So that's where the hydrogens can accumulate. And they actually flow back to the inside of the cell.
Where does it go for the gram-positives? Which space?
There's a little gap in between the membrane and the thick peptidoglycan layer. And so that's where the hydrogens go. So that was aerobic cell respiration, right? There was a final electron acceptor that was oxygen. But of course, we know that many microorganisms in particular are anaerobic, and so they don't need oxygen. So that means there has to be something else to pick up those electrons. So there are a variety of alternative electron acceptors. And in general, for anaerobic respiration, since it's not quite as efficient as aerobic respiration, cells can't make quite as much ATP. But if you're a single cell microorganism, that's plenty. So some examples of alternate electron acceptors: nitrates, sulfates, carbonates. Different microorganisms have different metabolic pathways. But let's go back and start summarizing things. So remember I said you need to know where things happen. So glycolysis occurs in the cytoplasm of all cells. The intermediate step is inside the mitochondria and of eukaryotes, still in the cytoplasm of prokaryotes. The Krebs cycle, also in the mitochondria and of eukaryotes, and in the cytoplasm of prokaryotes. An electron transport chain must be embedded in a membrane. So for eukaryotes, it's the inner mitochondrial membrane. But prokaryotes, it's just their plasma membrane, the cell membrane. The amount of ATP produced. We already know glycolysis is a net production of 2 ATPs. There's 2 more in Krebs for a total of 4, so not much. The amount of NADH, 2 in glycolysis, 2 in the intermediate step, 6 in the Krebs cycle for a total of 10. And FADH2, it's just 2 of those in the Krebs cycle. So 12 molecules of NADH and FADH2, and what are they carrying?
The electrons.
Electrons to where? Not there. To where?
The electron transport chain.
Electron transport chain. To make what?
ATP.
ATP, right? So, now this next table, these are numbers of ATPs. So all these numbers are ATPs. So again, we already know 2 in glycolysis and 2 in Krebs, and they're the result of substrate level phosphorylation and organic compound with the phosphate group transfers it to ADP to make ATP. But all the rest is made through oxidative phosphorylation. So the NADHs, remember we said 2 NADHs are produced in glycolysis, and they make 6 ATPs. 2 NADHs were made in the intermediate step, and they produce 6 ATPs. 6 NADHs produced in the Krebs cycle, and they yield 18 ATPs. So how many ATPs per NADH? It's not complicated math. How many ATPs per NADH? We got 10 NADHs, and they yield 30 ATPs. So how many? 3, right? So for the FADH2, we know that 2 molecules of FADH2 are made in Krebs, and those 2 molecules yield 4 ATPs. So how many ATPs per FADH2? 2. So why is it that NADH produces more ATP than FADH2? They're both carrying electrons, right, to the electron transport chain to make ATP. Why do we get a little bit more from NADH than from FADH2? Any ideas?
Is it because of more oxygen?
Nope, same amount of oxygen.
Is it because the FADH2 starts later in the cycle?
It is. So here we see the NADH is donating to the first complex in the chain. The problem with FADH2, it donates electrons later in the chain. And remember, as the electrons are passed from complex to complex, that causes hydrogens to get pumped. So if the electrons start at the very beginning, the maximum number of hydrogens get pumped. If the electrons start later in the chain, fewer hydrogens get pumped. And it's the number of hydrogens flowing through ATP synthase that makes ATP. So more hydrogens, more ATP. Fewer hydrogens, less ATP. So once again, the FADH2 donates its electrons later in the transport chain. So fewer hydrogens are pumped. So less ATP gets made. So does that make sense?
Yes.
So here we add up 38 molecules of ATP. It's usually 36 to 38 molecules of ATP. And what we're talking about here is the maximum theoretical yield if everything was perfect. And of course, we know nothing is perfect. So in reality, the maximum yield of ATP is something like 28 to 30 molecules for every glucose. But we're just going to use the maximum theoretical values. So why is it that prokaryotes average around 38 molecules of ATP, and eukaryotes average around 36. How come prokaryotes are a little bit more efficient?
Because they're just less complex altogether.
Well, they are less complex, and the main reason why is there's no mitochondria, right? So eukaryotes have to transport substances into and out of the mitochondria, and that takes a little bit of energy. But in prokaryotes, there's no mitochondria, so you're not transporting things around. And that's why prokaryotes squeeze out a little bit more ATP per glucose than eukaryotes can.
I have a question.
Uh-huh.
Is the maximum theoretical yield what it actually does or what it could be?
Ideal conditions when everything's perfect. In reality, it's nothing perfect. So like I said, the real number of ATPs is probably somewhere between 28 to 30, per glucose, rather than 36 to 38, because cells are not perfect.
Okay.
So that was aerobic cell respiration. The one type of anaerobic respiration we're going to talk about is fermentation. Since fermentation is very important for bacteria and other microorganisms as well as us. So again, there'll be a food molecule like glucose. It's going to be oxidized, but oxygen is not going to be used in this case. And since there's no electron transport chain, there's no Krebs cycle. So that means that ATP formation is limited to just glycolysis. So 2 molecules of ATP per glucose. If you're a single cell microorganism, you can live on that. But complex multicellular organisms cannot. And there will be some organic compound that acts as the final electron acceptor. And so usually what happens is that organic compound, it will accept those electrons and become reduced to produce the end product of fermentation. There are many possible types of end products. It depends on the organism and the environmental conditions. But lactic acid is a common end product, so acids are common end products. And that actually can be very beneficial. If you take Streptococcus, Lactobacillus, and Bacillus and mix them with milk, they produce lactic acid, which curdles the milk, spoiling it in a good way to make things like yogurt, buttermilk, sour cream, all because of microbial fermentation. Yeasts are single-celled eukaryotic microorganisms, and they produce ethyl alcohol and carbon dioxide gas. So your beer, wine, champagne, the alcohol is produced by the yeast, and the CO2 makes it bubbly. And if you mix yeast with flour and water and a little sugar, the yeast ferments and the dough rises. It rises because the bubbles of CO2 are trapped in the dough, and that causes it to rise. So fermentation has many useful purposes for us. But a wide variety of microorganisms ferment and produce a variety of substances, including some nasty things like acetone or isopropyl alcohol, which is rubbing alcohol. So when it comes to alcohol fermentation, the end result is ethyl alcohol plus carbon dioxide gas. With lactic acid fermentation, you can produce lactic acid only, which is homolactic fermentation, or lactic acid plus other end products, which is heterolactic fermentation. But in both cases here, ATP production is limited to just glycolysis, just 2 molecules of ATP. Now the NADH and the electrons that it's carrying are not being taken to the electron transport chain here. They're being taken to be used to reduce and produce the end product, whether it's lactic acid or alcohol fermentation. And that way the NADH and the NAD get recycled. And so as long as the cell has sufficient glucose, it can keep making enough ATP to survive. Now, so this works great for bacteria. Can human beings ferment? Is there any way that our cells can use fermentation?
Can they use lactic acid fermentation?
They can. Certain things like muscle cells, right? Your muscle cells are capable of doing that. So if you've ever gone to the gym and lifted weights, or you go jogging for a long distance, you've heard the expression, feel the burn. Well, that burn is lactic acid being produced in your muscles, and it burns. And so what's happening is that, you're working out so vigorously that you can't get enough oxygen to the muscles to make all that ATP. And so your muscles will briefly switch over to lactic acid fermentation. At that point, your muscle cells are only making 2 ATPs per glucose. That's not enough to sustain muscular contraction. And so you fatigue, you have to rest a few minutes or get more oxygen back to those muscles so they can start making a lot of ATP so you can resume doing work. So our cells are capable of fermentation for short periods of time, but we cannot survive on fermentation. We would die without oxygen. Within 4 or 5 minutes, we start dying. In the lab, we can test for fermentation. A common end product is acid. It's easy to test for acid with the pH indicator. In this case, the pH indicator is phenol red, which turns yellow with acid. We also have a Durham fermentation tube inside, a little bubble catcher, because some microorganisms also produce CO2 gas when they ferment, and we get a large gas bubble here. So acid and gas are characteristics we can use to identify unknown microorganisms. So this represents a biochemical test. You can add whatever carbohydrate you want to test, glucose, sucrose, lactose, maltose, whatever, and if the tube turns yellow, that's a positive test result. Now, just to mention it, you do know that sugars are not the only things used for energy, right? We can use fats and proteins. So you might recall, if you've taken nutrition, that, well, you tell me, how many calories in a gram of carbohydrate? How many calories in a gram of carbohydrate? Nobody knows? Nobody's taken nutrition yet? Well, it's 4 calories per gram of carbohydrate. It's the same for proteins, 4 calories per gram of protein. But for fat, it's 9 calories per gram of fat. So more than twice as many calories per gram of fat. And the reason why, if you think of carbohydrates, even complex carbohydrates, they're all simple sugars. They're all made of the monosaccharides, right? Just one subunit. And proteins are all made of amino acids, just one subunit. But fats, a triglyceride, it's got 2 components, glycerol and fatty acids. Each of these 2 components contributes to energy production. Glycerol can be used to make ATP, and the fatty acids can be used to make ATP. So that one molecule of fat has 2 components, and so 1 gram of fat gives you twice as many calories as either a gram of carbohydrate or protein. Because carbs and protein just have the one subunit, monosaccharides and amino acids. And that's the whole point to that. What's unfortunate about fat is that it tastes so good. We like to eat fat. So I mentioned a biochemical test, the carbohydrate fermentation. So we do use biochemical tests to identify bacteria, and we'll go over a lot of biochemical tests in the future. The idea behind the biochemical test is that you want to determine if an enzyme is present. The way you know an enzyme is present is by finding the end product of the enzymatic reaction. If the end product is being produced, then you know those cells must have that enzyme. So that's the point for biochemical testing, looking for enzymes. Just to mention it, we do know that photophosphorylation is a way to make ATP. There are some photosynthetic microorganisms which use light energy to make ATP. Here we're seeing some microorganisms growing in salt evaporation ponds. So these are roads, and so these are very large ponds. And the reason why they're colorful is because you have countless numbers of these microorganisms, and the pigment in their membranes are colorful. And when you get trains upon trains upon trains of them all together, you can see that. Now, these microorganisms are making ATP for themselves. They are not making any oxygen. So when it comes to photosynthesis, it's primarily the plants that produce oxygen. Almost none of the bacteria that are photosynthetic make oxygen. A small group, the blue-green algae, do make oxygen, but most microorganisms do not if they photosynthesize. So we can classify all forms of life based on nutritional type. So that depends on their energy source and their carbon source. A photoautotroph uses light energy and CO2 to feed itself. And here we see the cyanobacteria, the blue-green algae. So a type of prokaryote that does photosynthesize and make oxygen. But most other microorganisms that can photosynthesize do not make any oxygen. A photoheterotroph uses light and organic compounds to feed itself. Chemoautotrophs are the rock-eating microorganisms. They actually digest minerals. So they use minerals in organic chemicals and CO2. And the chemoheterotrophs, the largest, most diverse group of organisms, they use a wide variety of organic compounds for energy and carbon. So all the fermentative bacteria, all the animals, protozoa, fungi, and many other types of bacteria are chemoheterotrophs. So you can classify all living things based on nutritional type. And when you see the word auto, you should think of CO2, like what comes out of the back of your automobile, CO2. So any questions? Okay, we're running a little bit late, so we'll start lab about 5 after 1. Okay?
Got it. Thank you.
All righty.